from all over the world. My name is Li Ma. I'm the Senior Vice President of Alibaba Health. It is my honor today to host tonight's webinar. Today's session is organized by the Global MediXchange for Combating COVID-19 program, also known as GMCC. This program jointly established by the Jack Ma Foundation and Alibaba Foundation with the support of Alibaba Health to help combat the global outbreak of this COVID-19. This philanthropy program aims to facilitate online communication and collaboration across the borders, including sharing frontline medical experience, as well as latest R&D updates about fighting the pandemic. Even though the GMCC program has been launched for only 2 months, almost 1 million healthcare workers from more than 200 countries and regions have accessed the platform to learn more about COVID-19, and many doctors have had in-depth discussions. As the pandemic continues globally, we enlist the help of more experts to share on the platform their most current and effective experience to beat COVID-19. The virus does not distinguish between races and countries. And our knowledge, experience, and technology should also be shared if we are to win this battle against the virus. This is why we launched the GMCC program, and this is why we are here today. Because we believe this is one world, one fight. As the pandemic progresses, the development of effective treatment solutions and vaccines remains the focus of experts across the world. Today, we are very honored to have with us 5 world-leading scientists to discuss the latest research and development updates on COVID-19. I'm sure the following 1.5-hour discussion will provide us with much deeper understanding about COVID-19, as well as the progress of research on the virus. The experts we have with us today are Professor David Ho from United States. He's the founding scientific director of the Erin Diamond AIDS Research Center and the Clyde and Helen Wu Professor of Medicine of Columbia University, Vacla's College of Physicians and Surgeons. Professor Ho's research team has devoted considerable efforts to develop HIV vaccines, as well as antibody-based strategies and long-acting antiretroviral drugs as pre-exposure prophylaxis to halt or slow the spread of AIDS epidemic. We have Professor Paul Callum from UK. He's a professor of Virus Genome at Imperial College London and a vice president of infectious diseases and vaccines at Callum Limited. Professor Callum's research has identified how HIV develops resistance to antiviral drugs and has identified the first influenza disease severity gene in people hospitalized with influenza virus. Next, we have Professor Sharon Lewin from Australia. She is the director of the Peter Doherty Institute of Infection and Immunity. She is also the professor of infectious disease at the University of Melbourne and a consultant physician at the Alfred Hospital and Royal Melbourne Hospital. As an infectious disease physician and basic scientist, Professor Lewin's laboratory focuses on basic, translational, and clinical research aimed at finding a cure for HIV and understanding the interaction between HIV and hepatitis B virus. Professor Lewin leads the efforts on the Doherty Institute in the development of active and passive vaccination strategy testing to evaluate antibody neutralization and affinity, and the launch of a national adaptive platform trial for the management of mild to moderate COVID-19 disease. Professor Lin Qizhang from China. He's the professor and director of Center for Global Health and Infectious Diseases, Comprehensive AIDS Research Center at School of Medicine, Tsinghua University. Professor Zhang's team resolved the crystal structure of receptor binding domain of SARS-CoV-2 complex with receptor ACE2 and isolated several hundreds of antibodies from convalescent COVID-19 patients. The best antibodies are being developed for future clinical interventions. Last but not least, we have Dr. Daixing Zhou from China, who is our moderator today. He's the co-founder and CEO of Virus Genome Company Limited. Dr. Zhou is one of the pioneers of the new generation sequencing technology and non-invasive prenatal DNA detection technology in the world. He has provided a large number of professional solutions and experts in the field of scientific research and medicine for more than 10 years, and has greatly contributed to the development and application of the new generation sequencing technology in the Asia-Pacific region. At this point, I would like to hand over to Dr. Zhou to introduce the agenda for tonight's discussion. Dr. Zhou, please.
Thank you, Li. First, I would like to thank the Jack Ma Foundation, Alibaba Foundation, Alibaba Health for inviting me to act as the moderator for today's session. Good evening and good morning to you. Thank you for joining us today. We're now 5 months into the pandemic. Depending on which part of the world you are in, you may be experiencing different stages of the destructions brought by the pandemic. Shelter in place, business shutdowns, traveling restrictions, school closures, and of course, social distancing. The social and economical tolls to each nation are unprecedented. At the heart of the destruction is the fact that people are dying. Up to yesterday, the death toll, according to the number reported by Johns Hopkins University, stands at 310,000, and it's rising daily. There are a lot of efforts going on to find a cure, and sadly, we still don't have one. And equal sadly, there's a lot of misinformation swirling along the social media. Today, we want to bring to you the reliable and accurate information about the virus, the testing, and the treatments. To do this, we're very honored to have 4 distinguished speakers from the world. Again, they're Professor David Ho of Columbia University, Professor Paul Callum of Imperial College of London, Professor Sharon Levine of Peter Doherty Institute, and Professor Lin Qizhang of Tsinghua University. This is a true case of cross-border collaboration. Tonight, each professor will present his or her research for about 10 to 15 minutes, followed by 30 minutes of panel discussions. At the end of the session, if time permits, we will dedicate about 20 minutes to address questions raised by the audience. So without further ado, our first distinguished speaker is Professor David Ho. Many of you may recognize Professor Ho's work on treatments for AIDS patients. He's now leading a team funded by Jack Ma to look for a vaccine for SARS-CoV-2. Professor Ho, the stage is yours.
Thank you very much, Dr. Zhou. Good evening, good morning, everyone. Let me start by thanking the Jack Ma Foundation for organizing this webinar and for your support of our research. Since I'm going first, I will use a few slides to introduce the topic before sharing with you our current research efforts. We've been at this for only slightly less than 3 months. This audience knows well that the pandemic has affected virtually everyone on this planet. It began in China, of course, devastating the province of Hubei. And the world sat by and watched as if the rest of the world was spared. But very quickly, we recognized South Korea and Italy were being hit, and followed by Western Europe and United States in very dramatic form. As you could see here for the US epidemic, now accounting for 1/3 of the total caseload and about 1/4 of the death. Although the situation in much of East Asia and Western Europe and the US are becoming better, we see now wave after wave of the epidemic striking Russia, Brazil, and India. So there's no doubt that this is going to persist for quite a period of time. I think the clinical manifestations are well known to most of you, but let me just say that we know that a large percentage of infected individuals remain asymptomatic. And then for those who are symptomatic, 85% of the cases appear to be relatively mild. Approximately 10% are significant but not severe. And in our experience, about 5% of the symptomatic cases present with severe disease requiring not only hospitalization but intensive care. And the mortality rate is, the true mortality rate is probably around 1 to 2%, depending on the region. Although some official numbers, if you look at the WHO site or Johns Hopkins site, lists it as high as 6-7%. But that's probably due to inadequate sampling of the less severe cases. The measured incubation period had ranged from 3 to 6 days, with a mean of about 4 to 5 days. The reproductive ratio of the epidemic is 2 to 6, with a mean of about 3. The serial interval has been measured as 3 to 6 days, with a mean of about 4 to 5 days. The serial interval number being so close to the incubation period suggests much of the transmission occurs without symptoms. And we know the virus could persist, at least by PCR, for a couple of weeks. And then for those with severe disease, it initially was recognized as a pneumonia, but we now know it's much more than pneumonia. It's comprised of multi-organ failure, particularly affecting the kidneys. We know half of our ICU patients require dialysis. The disease also involved the heart, liver, brain, etc. And there seems to be a common feature of hyperactivation of the coagulation and complement systems. And then more recently, we recognize that there's a pediatric multi-system inflammatory syndrome that is probably post-COVID infection and is akin to Kawasaki's disease plus an element of toxic shock. So this is obviously a very, very severe problem. And as said earlier, we currently have no solution to address this except for the mitigation measures that are being taken. So my team started to work on this in late February with the initial funding coming from the Jack Ma Foundation. And given our background on development of drugs against HIV protease, we focus on the SARS-CoV-2 protease as a potential therapeutic target. And then we have worked on HIV neutralizing antibodies. So we thought we also focus on the spike trimer as our target for developing neutralizing antibodies. First, on the protease, my graduate student Sho Ikutani express and purify the main protease of SARS-CoV-2. We know this protease is required to cut at least 11 sites on the SARS-CoV-2 polypeptide. And without such cleavage by the protease, there would be no infectious progeny virions. And when the protease was purified, you could see the catalytic activity is shown here, pretty much giving us a KCAL-KM number that is reported in the literature. And for this, we use a synthetic peptide with a fluorophore and a quencher at opposite ends, and the cleavage would occur here. And this allows us to systematically screen a number of chemical libraries to find potential inhibitors. And I will simply share here that we are now working on 3 chemotypes that clearly block the catalytic activity of the SARS-CoV-2 protease at about 100 to 200 nanomolar concentration. So these are probably not close to being drugs. However, they serve as the starting scaffold from which to develop protease inhibitors. Recently, Sho and Farhad at Columbia crystallized these compounds into the soak the compounds into the purified protease and solve the structure. So this is a structure of the protease as had been reported previously with 2 catalytic sites, one here and one on the opposite side. And you could see we have one particular chemotype binding to the catalytic site. And then this 2 individuals involved also soak a second compound into the protease and solve the structure. And we know how these 2 chemotypes are interacting with the enzyme. We are now trying to solve the structure of the 3rd chemotype. But this is giving us clue as to how to proceed. This is sort of the active site of the enzyme, and you could see how each compound is embedded within the active site. But we could work with computational chemists at Wuxi AppTech to figure out how we could optimize the interaction between the ligand and the enzyme in each case, filling in more of the so-called S1 prime pocket as well as S3 and S4 pockets, and optimizing S1, S2 pockets. And so we realized that this is going to take many rounds of chemistry and optimization before we could improve the activity of these compounds by 50, 100-fold, and launch into drug development, true drug development efforts. So it's going to require a few months, but this is with only about 2 months work. This is pretty fast in our view. And I should say, while we're doing this, there's no doubt there are dozens and dozens of pharmaceutical companies that are chasing after the same with record speed. Let me just shift gear and talk about our work on the antibodies. This is an EM of a coronavirus, and you see the spike, and it's called coronavirus because it looks like the corona around the sun that had been observed by astronomers. And obviously, this spike sticking out is the target for neutralizing antibodies. So over the past couple of months, we have studied 40 patients to look at their serum antibody activity against SARS coronavirus 2. And here on the left side, we're looking at binding to the spike trimer for severe cases, and on the bottom, the binding to the spike trimer for non-severe cases. And one sees the curve shifted to the left for the severe compared to the non-severe, suggesting a much more robust response in terms of binding titers, probably by at least tenfold. Now, on the right, we're looking at the binding, not the binding, but the neutralization of SARS-CoV-2 for severe versus non-severe cases. And once again, one sees the curve shifted rather significantly to the left compared to the non-severe cases. So this led us to focus on the cases shown here, with titers ranging from 10,000 to 20,000 as the subjects for our isolation of SARS-CoV-2 neutralizing antibodies. And the process is schematically shown here. So we took 8 of our best cases, obtained blood, isolated mononuclear cells, and go through a multi-step cell sorting process. And I will just tell here where we gated B cells that are expressing CD20 and CD19. And you could see for this particular case, there's lots of B and the negative down here on the bottom are generally not found in a normal individual because these are the plasmablasts that are reacting to the acute infection. And then these are then further sorted for their binding for their phenotype of CD27, suggesting memory phenotype, as well as IgM. And once again, you see we have a number of cells. And once again, you see a preponderance of IgM, which is not typically seen. Again, evidence of acute infection. And then these are then further sorted for binding to the spike trimer. And here, for this particular case, you see it about 1% are positive here as compared to a normal. And it is from these cells that we then put into the 10x genomics and give pair antibody sequences from which we then construct the antibodies for evaluation for binding and neutralization. And we now have over 500 antibodies that are systematically being characterized for their binding and neutralizing properties. So I will just end by giving you a summary of where we stand of the monoclonal antibodies that we have tested so far. We haven't finished, but of those we have tested so far, half of them bound the spike trimer, about 15 to 20% are neutralizing, and half of the neutralizing antibodies are quite potent. And now we have a handful of candidates that have been identified against 2 different epitope clusters that we're considering as our development candidates. The top one, which here is simply labeled as MAB1, is no doubt directed to the receptor binding site, interferes with the binding of the receptor. And a second one, which is clearly not in binding to the receptor binding domain. And so we have these 2 as our top candidates in each category. And they are, we think, among the best in each class with neutralization potency as measured by IC50 in single-digit nanogram per ml concentration. and development as quickly as possible. And the antibody effort was carried out by my colleagues Li Hong Liu, Pengfei Wang, and Manoj Nair and Yaoxin Huang. So once again, thank you for the opportunity to present.
Thank you, Professor Ho. At such a short time frame, your team and yourself clearly make a lot of progress. Thank you. Our second distinguished speaker tonight is Professor Paul Callum from Imperial College of London. Professor Callum, you can start.
Thank you very much. And hopefully my screen is shared. Is that the case?
Yes, you're good.
Okay, thank you very much. And I'd like to thank the organizers and the Jack Ma Foundation, the Alibaba Foundation and Alibaba Health, and the organizers who've invited me along to speak today. I would like to spend just the next 10 minutes talking about the impact of genetics on combating COVID-19. And because genetics largely lends itself to larger scale collaborations, whilst many labs contribute to various aspects of genetics, it's only when you start to accumulate and federate the data and really share that some of the insights start to reveal themselves. So I'm going to touch on two parts of genetics. Three parts of genetics. They're based upon what we know about infectious diseases, both infections that exist, such as measles and seasonal flu, and then new incursions of otherwise known new pathogens into humans, such as pandemic flu, or now the one that we're here to talk about, COVID-19. And what we often observe when pathogens establish themselves and spread in a population are different relationships between age and the proportion of people that have severe or indeed fatal disease. And we know very well, for instance, that seasonal flu tends to be more of a problem for the very young and the very old, but as you reach late childhood and through to early old age, you are largely protected from the severity of seasonal flu. When we see a new flu come into the human population, this changes from a U to a W-shaped distribution, where unfortunately we uncover a lot of susceptibility in the otherwise healthy but middle-aged populations. And this is really different to, say, measles, that predominantly infects the young, or COVID-19, which is largely sparing the young, not exclusively, but largely, but is a problem in the older population and those with comorbidities. And the reason this is interesting from a genetic point of view is because this is really a reflection of three components of genetic variation: that of the virus, in this case a 30,000 base pair RNA genome; that of the host genetics, 3.2 billion base pairs of DNA; and then the adaptive immune system. And I've just put here the number of antibody variants that are likely to be generated in an individual, but also there's also the T-cell arm of the adaptive immune system. And how these three things interact often gives you your points on these epidemic and mortality curves. So I'm just going to take a few minutes on each of these to outline what is going on in the world and how some of the genetics that is already known is starting to play out. Starting off with the virus genome, this is something that my lab has been involved in for many, many years, and a lot of our work set up some of the frameworks for what is now going on in the world. And it's led to this view very correctly that individual sequencing is a fantastic thing to do, sequencing the whole genome. But when you federate these within a country or globally, that is when the true power of the virus genetics comes about. And the UK has taken this on board, as has many other places in the world. And the UK now, just the other day, reported that 16,380 genomes exclusively from the United Kingdom's epidemic have now been sequenced. There are many, many more of these in the world. And although this tells us individually what's happening in the United Kingdom at the level of different lineages, population expansion and contraction, it also tells us when you federate all of this information across the world, and an organization called NextStrain that is run out of Seattle and run by Trevor Bradford and Richard Nahar, starts to pull together subsets of these genomes to display the diversity of the virus as it spreads around the world, and enables us to tell some very important things very quickly. The amount of genetic diversity in the world is summed across all of these branches. And what you can immediately tell from tree structures like this is that there is genetic diversity. The virus is an RNA virus and it mutates. That's absolutely what you would expect. But there is no real structure to the tree that suggests that these sporadic variants that are generating here are under a selective advantage. And even though, and it was mentioned earlier in some of the introductions, there's a lot of chatter and sometimes misinformation around, there is no evidence at all that the virus is selecting variants that are fitter or better or dominating in the population at the moment, which is largely unsurprising because there is very little in the way of selective pressure, i.e. immunity, drug pressure, or anything else to drive the fixation of these variants. And the variation that we see is more akin to natural variation in the virus, founder effects, and the effects of different social distancing and lockdown procedures that will have an effect on constraining population growth. What this means is that we've moved very quickly over the last 10 years from sequencing hundreds of virus genomes in individual labs, particularly in genome centers, to thousands of genomes in small labs and the federation and analysis of this information. But it also leaves us some research questions and some practical questions about how we move this on further. And these are framed here, things that we're not able to do yet. Simple coupling of diagnostic RT-PCR to the genomes on one machine at the moment the sample is taken. Every time you break a chain between the diagnostic sample and the virus genome sample, putting the information back together again to link it back to an individual person and how they are reflecting the epidemic becomes hard. It's an information technology process. Seamlessly linking the virus genomes to metadata and epidemiology to make what you can see on those trees come alive is still tricky. And at least we forget that the reason this was found so quickly in China as a virus was because of the ability to sequence metagenomic sequencing in an agnostic way in hospitals, in the laboratories in Wuhan, and that should not be forgotten because our safety net for the future comes from having this same technology running, not in a research capacity, but in a surveillance capacity, probably in sentinel labs. So moving from the virus genome to the host genetics, again, the global effort on this is starting to marshal around things like the COVID-19 host genetics, where people are encouraged to federate and share information, and then individual countrywide efforts, such as one in the UK that is aiming to sequence 100,000 critical illness genomes. And the real point of doing the genetics now is the aim to be able to predict severity at the point of hospital admission, or to know somebody's individual relative genetic risk such that you could be prioritized for preventative or more aggressive therapies as they become available, or really to triage your ability to know who is going to require hospitalization or who's at risk of more severe disease once hospitalized. But there comes a sting in the tail with these sort of studies, which is they're very powerful in statistical terms, but to really leverage this quickly, you need to translate the genetics that you see in either the pathogen or the host into in vitro and in vivo biology models to understand the significance. And we were lucky in the pandemic influenza of 2009 to learn the rules of this. And one of the alleles that we discovered at the time for 2009 pandemic now seems to be coming back to be quite important, we think, in some initial studies that have started to emerge from Southeast Asia. And this was work that we and others did around a particular gene called interferon-inducible transmembrane 3, or IFIT-M3, in the flu pandemic of 2009. And to summarize an awful lot of work that was done in an equally short space of time, we were able to show that the removal of this gene in a mouse model turned an ordinary mouse, wild-type mouse, into a mouse that had fulminant viral pneumonia with relatively mild flu infection in the wild type and complete fatality just through the removal of this one gene. The same effects could be seen in tissue culture, in that if you remove this gene, the virus has an order of magnitude more ability to replicate. And most importantly, this gene is located on chromosome 11 in humans, and a single polymorphism in this gene was associated by us and others to be linked with more severe outcome of a respiratory infection. So most people in Europe have the T allele, most people, and the TT genotype is most common. In Southeast Asia, it's a more balanced, and you can see over here in rapid confirmatory work, in the Chinese population as a whole, this is the allele and genotype frequencies, where CC, the risk genotype, is about a quarter of the population. When Wang et al. Looked in H7N9 patients, you can see the increase in abundance of people with this particular allele and genotype. And then when you look at hard clinical endpoints, you can see that the CC genotype is required treatment with oseltamivir sooner, you have your steroids sooner, you're more likely to end up on a ventilator, and unfortunately, you're more likely to succumb to the infection as opposed to the TT allele. In the European population, within 64 base pairs of this mutation, there's another mutation that is the mirror image of the CC allele, so that the European populations also have a risk in this genotype. And this work is now starting to show that this allele may well have similar predictive power in a small study of patients with severe illness in China, and it really needs to be looked at quickly, and this is something that my lab is involved in now. And just finally, I'm not going to touch on this for very long because Professor Ho touched on this so well, but we and others are looking at the ability to immunize mice that have human immune systems as a analogous way of finding antibodies that have previously been pulled out of humans. It has one advantage, I think, which is by it being an immunization strategy, we can focus the immune response on the spike gene and maybe explore other activities within the binding motility of antibodies that are perhaps rarer in humans. And the humans are looking at many of the other coronavirus proteins, but we already start to see, as Professor Ho pointed out, all of the different binding patterns. The real trick now is to work out how we can manufacture these quickly, shorten the process that normally takes 2 to 3 years to get to a clinical trial, and how we look for synergy, such that adding more than one antibody together can give us a order of magnitude, perhaps improvement over the individual antibodies, because we have one problem with these antibodies, unlike treating HIV, which is what we load into the periphery, the peripheral blood of a human, needs to be able to translocate and make its way into the lung, where this protective or therapeutic effect is most likely to be important. And it's really understanding that immune response that we need to do now, the ability to pull antibody repertoires apart. And this is an example from malaria, but it shows you the same phylogenetic trees that we use for viruses can now be used to describe the immune system of an individual as they develop their protective immunity to a vaccine or to their natural infection. And what we've recently been doing in terms of review of the literature, the understanding of the natural cause of infection from people with severe disease and have a good antibody response, those with milder disease that perhaps have a weaker antibody response, how long this lasts, and why it declines over time, and are there any particular ways that we can boost this and follow it, both in molecular detail, but also at a serological level. So just to close, unfortunately, I think many of us on this panel have been working in this and allied fields for many years now, and every time a new virus comes along since my lab started to work in this area, I've updated this slide. This slide is now full, and moving on over the next few years, I will need to move on to slide 2. But it's just a reminder that whilst we're all focused on SARS and COVID-19, there's a periodicity of new viral infections into humans. This won't stop just because we're looking at COVID-19 and SARS-2. Nature will keep having these incursions, and our guard against all of these other pathogens needs to be maintained, and cross-learnings between these will advance us quickly in COVID-19. So thank you. I'll leave it there.
Thank you, Professor Callum. I'm very delighted to see so many sequences being sequenced and shared around the world. This is something probably not been done in history. So this is great. And I definitely going to have my IFITM 3G sequence tomorrow. Now, let's move to our third distinguished speaker tonight. Professor Levine will be our third speaker. Professor Levine, the stage is yours.
Hello?
Professor Levine, can you?
Please unmute your microphone. Professor Lewin, please unmute yourself. We cannot hear you.
Yeah.
Hello?
Your microphone is.
Your microphone is on mute.
Back to Professor Callum.
Michael, could you switch to Dr. Lewin's menu, please?
And just to check, it's Paul Callum here. I have unshared my screen, I believe, so I don't think I have control.
Yeah, if you just stop sharing your screen, that'll be fine. Yeah.
Yeah.
Hello, Professor Lewin, we cannot hear you.
Presentation is not popular.
Should we move to the next person first?
Hold on for 1 second.
She's muted. She's muted.
It's still on mute, I think, right? Professor Lewin is still on mute.
Okay, while we're waiting, how many of you have your IFITM gene sequenced? It's very important now.
Yes, as we're perhaps, I mean, it's clearly early stages in this, and we need large-scale association trials. The biology for influenza is pretty solid, and we need to work very hard to see if it is just influenza or whether there's a role in SARS-2 as well. But yes, it will be interesting to see how this area develops quickly. And I think just to fill some time whilst you're getting the other people up, it was really that sharing between what we were doing in the UK and the US, publications, and rapid replication and extension of the work in China in 2012 and 2013 that moved it on at a pace that you couldn't do apart from sharing data. So I think we are in the same place now with COVID-19, and I hope to see more of this in the future.
I think Professor Lewin is still cannot connect, so maybe Professor Zhang can go ahead first.
Yeah.
Yes.
So Professor Zhang, you're next. Can we
put up the slides?
Right, we're doing that.
Okay, before I start, I would like to thank the Jack Ma Foundation and Alibaba Foundation and Ali Health for inviting me to present our updated research progress. My name is Lin-Qi Zhang from Tsinghua University School of Medicine. I'm going to talk about the human neutralizing antibodies to SARS-CoV-2, the path towards clinical interventions. Thanks for the previous speakers, laid a great foundation for the introduction of antibodies. So my presentation will go faster than planned. Okay, as Professor Ho has already shown you, this is the coronavirus under the electron microscope. So there's some delay in the presentation. So what you see on the screen is the look-like of coronavirus under the electron microscope. As we are aware now that the coronavirus exists in many different animal species. On the left is from the pigs, on the right is from the chickens. You can see they all share the similar phenotypic looking. That's why they're all called coronaviruses. The new coronavirus isolated from humans, SARS-CoV-2, looks very similar. And then the spikes on the surfaces are the protein that play a critical role in mediating the viral entry into the target cells. And that's the target that we have been focusing on. And since the outbreak of COVID-19 diseases in Wuhan, China, my group and the Professor Xinqian Wang's group from Institute of Life Sciences has been working together, try to understand the structure basis of the viral protein interactions, spike protein interaction with receptor ACE2. So by understanding the structural details, we were not only able to understand how the virus gets into the cells, but also providing a precise target for developing agents to block these interactions. So on the right really highlights the receptor binding domains interactions with ACE2. In the middle is the molecular structure, the crystal structure of highlighting the interface between the ACE2 and the receptor binding domains. There's some little weird thing about this mouse. Do you have a mouse pad? Do you have a mouse pad? Mouse pad? So as you can see, that's okay. That's okay. The panel on the right really highlights this particular receptor binding domain's interactions with receptor ACE2 in the context of the trimeric spike protein. So our team and other team in the world has been working around the clock to identify the agent that are capable of blocking this interaction between RBD and ACE2, thereby to block viral entry. So we have been working on the antibodies aiming to block these interactions. As we know very well, that once the human body is in host infected by virus, our immune system produces tremendous number of antibodies against viruses, in particular against surface protein, in this case, spike protein. But only few of those antibodies are capable of blocking viral entry. So we name those antibodies as a neutralizing or potent neutralizing antibodies. Isolating, characterizing those antibodies are the major focuses of entire field, because antibodies not only able to serve as antibody drugs to treat and prevent infection, but also serve as a guide for our future vaccine design and development. On the middle and right really highlights the structure of antibody that's circulating in the blood. This antibody constitutes about 20% of plasma protein, so constitutes a tremendous amount of our protein in the plasma. It looks like the spring shape that the children play in the field. And the the specificity of the antibody come from the both ends of the branches. The role of neutralizing antibodies has been demonstrated in many different cases, in both clinical and in the laboratories. And as we learned from the multiple anecdotes as academic publications as well as the media coverage, we know there's ongoing plasma therapies that are going on to treat severe cases, largely due to the neutralizing antibody exists in those recovered plasma. However, due to the various reasons, there are some drawback with this strategy because of limited resources and hard to standardize and maybe co-contaminated with other infectious agents. So isolating the potent neutralizing antibodies from those patients will become a major focus of the field. So how do we isolate antibodies from these recovered individuals? And the next slide highlights overall a procedure. Basically, you started with a convalescence of recovered patients and collect a minute amount of plasma. This could be as little as 10 mL or to 5 mL, even 1 mL. Using a sophisticated sorting technology that highlighted in the upper right, and then throw in the viral specific proteins, in this case, either the spike protein of COVID-19 virus or the receptor binding domains. You could basically sort out the antigen specific B cells that are encoding the antibodies on their surface. Through the single cell PCR amplification, you could amplify the heavy chains, the light chains of the corresponding antibodies, and through the in vitro transfection, those antibodies could be produced in the cultural flax. And then you could test those antibodies and characterize those antibodies in terms of binding, neutralizing activities. And then you select the best of those to treat patients and to prevent. So from the 8 recovered individuals that we selected during the early outbreaks in China that enrolled into the Shenzhen #3 People's Hospital, we are able to isolate 206 antibodies that targeted to the specific proteins, receptor binding domains on the spike proteins. Here we show the 3 selected represented patients and their genetic characterization. As you can see, the boxes and the ovals that highlight those B cells are able to bind to the receptor binding domain of spike proteins. And then once we have those genes sequenced, we could characterize their genetic features. As you can see, they highlight in different colors, representing they came from a diverse genetic background. Diversity and distribution, there's a big degree of variations among different patients. Once you have these genetic features and genetic sequences, and then you could put them into the cultural flax and to produce those antibodies and characterize their binding and neutralizing activity. Those highlights those antibodies that really able to bind to the receptor binding domain that we put into as a bait to fish those B cells out. Those highlight in red are the antibody able to bind the receptor binding domain with exceedingly high binding activities. And those ones are we selected to characterize further in terms of their neutralizing activity against virus. So you can see there's great variability from patient to patient. I have to say patient 2 and patient 5 are severe cases and consistent with the Dr. Ho showing that severe cases tend to have a higher level of not just the serum neutralizing antibody, but also B cells that are able to produce those antibodies. So once you have a protein form of antibody, you basically analyze their antiviral activity. How you actually do it, you mix with antibody and virus and put them into the cell culture. So the slide is really showing the spots. Each circle, the spots in each circle represent a viral foci. So as you continue to change concentration from high concentration from the left all the way to the right to the low concentration, you could see the number of viral foci increases. And then this change pattern allows us to evaluate the neutralizing activity of those antibodies and select the best ones that are able to inhibit viral replication at a very low concentration. And then you summarize those numbers into a curve, and that really gives you a very clear indications how different those antibodies in terms of inhibiting those COVID-19 virus infections. As you can see, the curve highlighted in red and purple on the left really indicate they are much higher in terms of capacity in inhibiting viral entry. The panels on the left represent their antiviral activity in the setting of a pseudovirus entry assay. The panel on the right really highlights their neutralizing activity against live SARS-CoV-2 virus. Basically, they are consistent, showing those antibodies are really potent in inhibiting viral replication. To further analyze their structure basis for the antibody that inhibiting, showing those inhibiting activity, Dr. Wang's team and I have collaborated to solve the crystal structure of the antibodies. And then on the left, you can see this highlights the antibody highlights in the purple and yellow, how that recognized RBD will interrupt the interaction between the color CN and ACE2 receptor in green. So since the interactions between RBD and ACE2 is critical for viral entry, the intercept, the binding of this yellow and purple color produce this spatial interruption of this proper interaction, thereby preventing the viral entry. So this is one of the antibodies that we isolated, demonstrating a very potent activity against viral infections. In the middle, highlights one of the control antibody that failed to neutralize the viral entry and viral infections. As you can see, this antibody really comes in from the sideways, doesn't have any impact on the proper interactions between the viral receptor binding domains and the receptor ACE2. So since the COVID-19 virus, the spike protein exists in the trimeric form, so we superimpose the left panel antibodies, a crystal structure onto the overall trimeric background. As you can see, this antibody not only able to bind to the receptor binding domain in the upper position, but also in the down position, suggesting this antibody has potential to have a full coverage of spike protein on the virion, thereby providing potent antiviral activity. So what you do once you have those antibodies, basically as Dr. Ho and Dr. Callum said, once you have those antibodies, you put into a mass production through the fermentation and purification. So, and then once you have those procedures settled, you need to test it in the safety, efficacy in animals before you move into humans. So we have been collaborating with a biotech company called Imperial Biosciences to rapidly develop those antibodies for clinical use. Lastly, I would like to thank our team members. Without them, I would not be able to present those exciting discoveries. On the left is my group from Tsinghua University. In the middle is led by Dr. Zhang Zhen from Shenzhen No. 3 People's Hospital. On the right is the Professor Xinchen Wang from the Institute of Life Sciences at Tsinghua University. Thank you.
Well, thank you, Professor. That data is very fantastic. I'm very excited. Looks like both your work and Professor David Ho's work indicates neutralizing antibody looks like very powerful. Looks like something we're going to have a new weapon against the coronavirus. Now, our final speaker. Finally, we solved the technical issues. Final speaker, Professor Sharon Levine, will give the final presentation. Professor Levine, please go ahead.
Thanks very much, Dr. Joe, and apologies about the earlier events. My Zoom somehow crashed on my computer, clearly overuse. It got a bit tired, and so I'm on my phone at the moment. So if I could get you to put up my slides, that would be great, because I won't be able to do it from here. So if someone could put up my slides, that's fantastic.
Okay, we are doing that.
Okay.
Great. If you just pop it on slide view, that would be great. Thank you.
Slides on.
Yeah, the slide is on.
And could you just put it on? Okay. Could you just put it on the slide view so we can see the full screen, the slide on the full screen?
Slide view.
Yeah.
Full screen.
Yeah, it's a bit of delay. Hold on for 1 second. Yeah, full screen, please,
Oh, okay.
Michael.
Sorry. Great. Thank you very much. Okay, well, it's a great pleasure for me to be here as well. I'm speaking to you from Australia, and I'd like to thank Dr. Joe for the kind introduction, and also thank the Jack Ma Foundation and the Alibaba Foundation Healthcare for hosting today. And again, I want to also thank the Jack Ma Foundation for their support of work at the Doherty Institute. At the Doherty Institute, we were the first to isolate SARS-CoV-2 outside of China and share that widely on January the 29th. And that led to us meeting with the Jack Ma Foundation and their support for our vaccine program. But tonight I've been asked to speak about COVID therapeutics and an overview of that area, some work that we're doing in Australia, and I'll briefly mention some of the vaccine work as well. So if I could have the next slide, please. So this is the epidemic curve of what has happened in Australia, and you can see that we've had a very small outbreak compared to the rest of the world. It shows you the origin of cases in Australia. In purple are people diagnosed with SARS-CoV-2, showing you that they had a travel history, and in blue is people that acquired it locally, and green, unknown source. And in context of what is happening globally, these are really tiny numbers. If you could press the next slide, please. So as of yesterday, there's only been 7,000 cases in Australia, under 100 deaths, and over the last 3 weeks, less than 30 cases per day. And that's very much a testament to the public health response in Australia, with high levels of testing, about 1 million tests in a country of 25 million people. Our positivity rate is at about 0.7%, and our testing rates are high, about 4% of the population are currently being tested. Next slide, please. So many people in this room and in this seminar will have far more experience with treating COVID-19 than we've had in Australia, but I'll talk to you tonight about the rationale and timing of treatments for COVID-19. Next slide. A bit about the antivirals and what's available when they're used. Next slide. The results of some recent large randomized controlled trials. Next slide. Some outline of some clinical trials currently happening in Australia. And next slide. And finally, a little bit about the Jack Ma vaccine program at the Doherty Institute. I won't go into great detail about that tonight. And next slide, please. I think this slide summarizes very nicely the distinct clinical phases in COVID-19 and our emerging understanding of the pathogenesis of disease. So in general, in the first week of illness, as many people will know, there's a largely mild infection. In the second week is when people often would become unwell. And third week, when people are seen to become severely unwell, and that's when deaths occur. And similar to SARS-1, there's now an emerging understanding that the first week or 10 days of the illness is characterized by viral replication, and the second phase of the illness characterized largely by immune dysfunction. Next slide. And this very much, I think, is now influencing our understanding of when treatments might work. So is that antiviral therapies or strategies that target the virus may well have their best chance of working early in disease, while immune modulating or immune suppressive agents would have the best chance of working in the setting of severe disease. And next slide. And here I've just summarized some of the available therapeutics that are targeting the virus, either antivirals or antibodies, as you've heard about from the previous speakers. These antivirals can be directly targeting the virus itself, and I've listed a number of the antiviral agents currently under investigation, or could be targeting host proteins which will affect viral replication, agents such as hydroxychloroquine, chloroquine, or interferons. Antibodies are yet to be evaluated in large-scale trials that can be evaluated as convalescent sera, hyperimmune globulin, or now great hope for these designer monoclonal antibodies that Lin Shi and David described earlier. Next slide. Once people progress to more severe markers of disease, suppressing the immune response is largely the basis of strategies, either blocking IL-6, blocking IL-1, or blocking other inflammatory pathways such as the JAK-STAT inhibitor baricitinib. And over the last few weeks, I think it's become more apparent that managing complications might be key to managing severe disease, particularly complications of the thrombotic arms with anticoagulation, thrombolysis, or even intravenous immunoglobulin or aspirin. Next slide, please. I like this slide because it tells you how much is going on and that it's slightly chaotic, the amount of clinical trials that are occurring around the world. And understanding and comparing these clinical trials really does become very challenging for clinicians. And I'm going to try and just focus on clinical trials that have recently come out that are giving us a slightly more definitive answer or understanding of the best approaches to treatment. Next slide, please. So I'll start with remdesivir. I think many of you will have heard of remdesivir, an antiviral agent that blocks the RNA-dependent RNA polymerase developed by Gilead. There are now 7 large randomized clinical trials, either completed or underway globally. And recently we heard the results of one clinical trial, if you could press the slide, please, which is a randomized study coming from China. And those results have been published in The Lancet, and I'll discuss those briefly. And then 2 other press releases around clinical trials, one in a large adaptive clinical trial from the US and another from Gilead, comparing 2 different duration of treatment, 10 days versus 5 days. Neither of these clinical trials yet are published. Next slide, please. So these are the results of the only randomized clinical trial currently published, performed in China, double-blind multi-center study of remdesivir given for 10 days. And the primary endpoint was time to clinical improvement out to day 28 using a 6-point ordinal scale of severity. The study enrolled 237 patients, but was stopped prematurely due to declining enrollment. And you can see here on the left a comparison of viral load in the nasopharyngeal aspirates of remdesivir in red, or the control arm in blue, showing really no distinct antiviral effect. And in fact, there was no clinical benefit of remdesivir. This was, of course, disappointing, but a small study, but a very well-designed and well-executed study. As I mentioned earlier, there was some excitement around an announcement, but not yet publication, of a large platform trial led by the NIH called ACT. And this apparently showed a reduction in time to recovery from 15 to 11 days with 10 days of remdesivir. This was a much larger trial of over 1,000 participants, and the details of the participants are yet to be announced. There is, however, some great hope with remdesivir. It's probably acting as a mild antiviral drug and will likely be needed to be combined with other agents. Next slide, please. The other clinical trial that I think has generated quite a bit of excitement comes from Hong Kong, and it's the first effective demonstration of an effective antiviral regimen when used in combination. This was an open-label randomized study of Kaletra, an HIV protease inhibitor, compared to Kaletra coupled with ribavirin and interferon beta. And the idea for this combination came from not only in vitro activity of Kaletra, ribavirin and interferon beta, but prior experience with SARS-1, specifically in Hong Kong. This study was designed quite differently, and it's really important to think about the study designs when trying to interpret these. Here, the primary endpoint was virological, or the time to a negative nasopharyngeal swab. It's also really important that the participants in this trial had far shorter duration of symptoms than in most other published trials. The median duration of symptoms was only 5 days. So treatment was occurring in that period that I showed you earlier where there's viral replication. And very excitingly, you can quite easily see on the left a clear effect of the intervention with a greater reduction in viral load at all time points measured. There was also a shorter time to resolution of symptoms. I think this is a very different population to many of the clinical trials that have been presented, that have been published already. However, it does show that interventions that are starting early, potentially at less than 7 days, targeting the virus can not only reduce viral load, but lead to enhanced clinical outcome. Next slide, please. So if I could just summarize some of the take-home messages from many clinical trials, both randomized and non-randomized studies. Timing of initiation of antivirals appears to be important, with effects best seen when given within 7 days of symptom onset, and there's been other clinical trials suggestive of this. Next slide. The inclusion criteria are very important when trying to interpret efficacy. There's a very wide spectrum of clinical presentations driven by different underlying pathogenic mechanisms, and therefore the treatments will differ at different stages of disease. Next slide, please. The primary endpoint is also important. These vary greatly from time to recovery, death, or surrogate virological markers. And final point on this slide, please. And antiviral drugs to date really only show very modest effects. Combination antivirals certainly look for far more promising, and certainly that's been the case from other viruses successfully treated, such as HIV or hepatitis C, and high hopes currently resting on antibody therapeutics, not yet evaluated in large clinical trials. And next slide, please. So I'm just going to talk about 2 large randomized studies currently underway in Australia. One is ASCOT, the Australian COVID-19 trial, led by Stephen Tong, a clinician scientist based at the Doherty. This will look at adults with COVID-19 who are hospitalised but not in intensive care, and enrolment will require less than 12 days of symptoms. It's a platform trial. Currently, the plan is to randomise people to hydroxychloroquine versus Kaletra versus the combination of hydroxychloroquine and Kaletra, but this may well change as new evidence comes in. The advantage of an adaptive trial platform, the control will be compared to standard of care, and the primary outcome here is advanced respiratory support-free survival at day 15. Next slide, please. The other large platform trial being currently undertaken in Australia as part of an international study. This is an interesting trial called REMAP-CAP. It's a randomised, embedded, multifactorial adaptive platform trial for community-acquired pneumonia. And it actually started with the planning to adapt in the event of a pandemic. It is enrolling adults with COVID-19 in intensive care. It started several years ago and was enrolling individuals with pneumonia, but has now adapted to also include COVID-19. It's currently active at 180 sites, 15 countries, and already 470 people enrolled in COVID domains. And the primary outcome here is a composite of in-hospital mortality and days alive and out of intensive care during the first 21 days. Next slide, please. Because it's a platform trial, it's able to incorporate a number of different domains. And you can see here, there's several questions being asked embedded within REMAP-CAP, the use of corticosteroids, the use of macrolides, the use of antivirals, immune modulation, vitamin C and heparin. Within this framework, each of these interventions can be rigorously assessed. And there's enormous benefits for having a platform trial, particularly one that was up and running prior to the onset of a pandemic. And it's a great lesson, I think, to many of us in clinical research about the importance of preparedness of pre-approved protocols. If you just move to the final slide. I've spent the session talking about therapeutics, given the focus of the other talks, but I just want to briefly mention some of the vaccine strategies being pursued at the Doherty as a result of the support of the Jack Ma Foundation. These include active vaccination strategies. If you press the next slide, please. Using protein-based approaches, or particularly a clamped spike protein developed by the University of Queensland with funding from CEPI, but we're performing these vaccine studies in partnership with University of Queensland, CSIRO and the Doherty, evaluating the effects on neutralization. And next slide, please. And a separate program using adjuvanted virus-like particles that express the spike protein. And the next slide, please. And similar to what we heard before, an approach to passive vaccination, identifying antibodies that bind tightly to the receptor binding domain using either nanobody libraries or B-cell clones from recovered patients. A similar approach to what was described by David and Lin Shi earlier. And next slide, please. And I'd like to acknowledge the team that are part of the Jack Ma Foundation, the leadership of the ASCOT trial, Steve Tong, the leadership in Australia of REMAP-CAP, Steve Webb, support from the Jack Ma Foundation, as well as many other funders that have supported our work on COVID-19 at the Doherty Institute. Thanks very much.
Summary in a very confusing world of treatments. Now, all the speakers have finished their presentations. With the session today, we'll move into the panel discussions. Professor Lewin, I think you are very late in your time zone. Let me start with you first. You just summarize all the complicated and confusing world of the treatments. And I know you face patients almost every day these days. If I'm physician today, which I'm not, and facing a COVID-19 patient, what should I do these days? What kind of drug should you recommending you to treat a patient today?
I think at the moment, what I have shown you is that there is really insufficient evidence for specific antiviral treatments. And our recommendations in Australia are that patients should be best to be enrolled in clinical trials to try and address this question. There just is insufficient evidence of activity of the drugs that have been tested so far. Now, one intervention that may be different is remdesivir. At the moment, remdesivir is not available in Australia. And I personally would like to see the results of the encouraging trials from the platform studies led by the NIH before I was proceeding with using remdesivir. From the studies that we've actually seen, we can't see any clinical benefit. So my approach is not to use a specific antiviral treatment, to of course use supportive therapy as needed, but the idea is to have patients enrolled in a clinical trial. That's why within Australia, even though we've got so few cases, we've been very keen to establish this national network. The ASCOT trial will be in 86 sites across Australia. And that means that should we have a resurgence of cases or a second wave, we're just relaxing restrictions now, we'll be well prepared to enroll nearly all individuals in a clinical trial. So my approach would be not to use a specific antiviral and to be enrolling patients in clinical trials.
Okay, that's great. But there's only limited number of trials going on, and there's so many patients around. So without the trial, any suggestions for the physicians to follow?
Yeah, I mean, the best approach, the best approach is to use supportive care, of course, fluids, oxygenation, ventilation, treatment of secondary bacterial infections. Using an antiviral drug with unproven efficacy doesn't mean that you're doing something good for the patient. All drugs have side effects. We know chloroquine, hydroxychloroquine have side effects. Kaletra has side effects. Remdesivir has side effects. And I think to use a specific antiviral, you need more evidence of efficacy over adverse events. And we're just currently in a position where we don't have that evidence.
Okay, thank you. Second question goes to Professor Ho. Professor Ho, you are famous for cocktail therapies for AIDS patients. And in this pandemic, there's some indications, at least there's a recent article on triple combinations of 3 different drugs to treat a COVID patient. Do you think a cocktail therapy may work for COVID patient?
I don't know the answer to that question at this moment. The triple combination that you're likely referring to was covered by Sharon Lewin just now, with lopinavir, ritonavir as one drug, beta interferon, and ribavirin as the third drug. Out of Hong Kong, showing greater suppression of SARS-CoV-2 in the treated patients. I do know that lopinavir, ritonavir has absolutely no activity against SARS-CoV-2 in our hands. It's not a protease inhibitor for SARS-CoV-2, and it has no effect on virus replication in the laboratory. So I'm not inclined to use such a combination, although ribavirin and interferon have long been touted as antivirals. So they are possibilities. I think what we're doing in the U.S. is to, at face value, trust what the NIAID results are showing us. So remdesivir is, I would say, a baby step forward. It's sort of like AZT was in 1987. It's slightly helpful. And when given such a weak drug, we need to build on that. And so I would support the idea that we do clinical studies, not just, do whatever each individual physician prefers to do, but conduct clinical trials so we can learn. And then there are agents we could use to add to remdesivir. And I think many such trials are being tested. Beta interferon could be added to remdesivir today and be tested in a control study. And there are several other agents that are known to have activity against SARS-CoV-2. They will be so that's what I would recommend. But I would say that overall in China and in the U.S. get the neutralizing monoclonals out very soon. For example, in the US, Regeneron here in New York will likely get their antibody into human testing next month. There are many others that are trailing behind just by a little bit. Our own antibodies, we started much later, but I think we have the possibility of getting them tested in humans by late summer, if all goes well with the development process. So I suspect within the next 6 months, we're going to have numerous candidates in clinical studies. And sounds like many of them are very much the same as Dr. Zhang described. They're all largely directed to receptor binding domain. That's true of the Regeneron antibodies. It's true of Dr. Zhang's antibodies and true of one of our most potent antibodies. But now I think we will see another class come up, and whether they are useful in combination or not, we have to wait and see. So I think the area is very promising over the next few months. And this, compared to plasma therapy, has the advantage of being able to manufacture in quantities safely and without risk of contamination from blood pathogens. And more importantly, we could isolate only neutralizing antibodies and avoid non-neutralizing antibodies, and to minimize the risk of what's been called antibody-dependent enhancement. So these are just some thoughts I could share.
Professor Zhang, your comments?
Yeah, I would agree completely with Dr. Ho's assessment. I think antibody provide a very exciting alternatives or complement to the antiviral drugs. And then since the epidemic started early in China, so we took advantage of involving the patients into our study. So we are a little earlier than other groups in terms of isolating antibodies and put them into the development process. As Dr. Ho pointed out, competition, there's multiple groups that in the world are working in the same area, including Dr. Ho's group. So we are working as hard as we could to push those into clinical development. Hopefully, we'll show some promising results, not just for treatment, but also for prevention.
Great. We look forward to it. So now let's move to the topic of vaccine development. So globally, I think everyone's eyes now is on vaccine development. And there's a lot of efforts being put into developing one. So how many different methods being used to try to make an antibody, and what stage are they? Professor Zhang, can you give a summary on?
Vaccine is really one of the driving forces for the COVID-19 prevention, because many vaccines are aimed to develop for prevention purposes. As we know, there's multiple strategies that are being tested. There are front runners in terms of entering clinical trials, such as mRNA-based vaccines, the vector-based vaccines, and also some of the acute vaccines. But we're still in a very early stage. It's very hard to tell at this stage which one will provide the safe and effective, and also scale up and then accessible to larger populations. But given what we know from the animal studies, many of those strategies are able to induce good amount of immune responses. But whether those responses will correlate and will be the surrogate for protection need to be studied. So overall, we are still in the early stage to answer that question. And hopefully some of the more type of vaccine will put into clinical trials. So we could say more definitively based on the data, not by our just hopes.
Thank you, Professor Zhang. Professor Ho, you are also working on getting a getting a vaccine developed. How far along is your program?
Actually, our only work in vaccine is the passive antibody effort. We're not working on active vaccination. We have a few ideas, but at the moment we have not launched those ideas.
Okay. Professor Lewin, how about vaccine developments in your line of work?
Well, I think the most advanced strategy is the approach that we're doing in collaboration with University of Queensland, Paul Young and his group, where they have a clamped trimer protein that they originally developed for MERS and rapidly pivoted to SARS-CoV-2 once the sequence was announced. And those clamped trimers have been tested in mice and show evidence of inducing neutralizing antibody. And the plans are to move into phase 1 studies in June. And so that's moving along quite quickly. That's using a standard protein-based vaccine that does induce what looks like potent neutralizing antibodies. The other approach that was being used, led by Dale Godfrey and Joe Teresi at the Doherty Institute, is using viral-like particles with the spike protein on the surface of those particles. They're still in animal models to assess immunogenicity currently.
Okay, so looks like it's still a bit away. It's not, now we're going to get it very soon, right?
I think clinical trials in June is pretty good, actually. That doesn't sound too far away to me. The University of Queensland, CSIRO, the Doherty Institute, I think that's a pretty great effort, actually. So at the moment, there's 10 clinical trials happening, as Dr. Zhang said earlier, with DNA vaccines and RNA vaccines and other vectors. You know, I think that the challenge will be safety, to ensure that these are really truly safe, and of course efficacy, and doing it in such a short period of time. But I think the progress of moving these agents into clinical trials so quickly is actually pretty remarkable.
Great, thank you. The final question for the panel discussion is to Professor Callum. Now, as we know, many parts of the world are entering the phase of reopening, for which social distancing and testing are essential. So testing in UK has gone through multiple rounds of the debates. What's going on there, and what do you see globally if we wanted to open up? How broad we should do the testing, and testing, is testing so essential to that? Yeah.
Well, I'm one that does believe testing is essential. I think the ability to test and trace and action is critical. I think what you see globally are the problems of different size populations and countries. In countries where your population size is in the order of 10 to 20 million, your ability to deploy a large number of tests and cover a large amount of the population theoretically with testing is a hard undertaking, but not impossible. When you try and then apply those same principles nationally, as we do in the UK, where we have a population of 66 million, or in the States where there's 370 odd million people, that ability to scale centrally becomes really, really hard to do. And what you've got to do in those situations is, in my opinion, look more locally, to think about regions, to think about how you test within cities and regions of a country, rather than trying to have a very coordinated mass testing system. The trick for all infectious disease outbreaks is speed. Speed. is the one thing you should remember all the time. The speed from sample to laboratory, from laboratory to reporting, and from reporting back to actioning. Every time you have a delay, it reduces the effect that you can have. And that again argues for scaling your testing to be the right size for your population at hand, and thinking locally as well as nationally. So yes, it will change, and that's for the diagnostic testing. When it comes to antibody testing, well, a lot of work is going on there. We've now got major diagnostic companies rolling out their N-gene antigen test for detecting antibodies to the N-gene. So that's going to be very interesting to see levels of seroprevalence. But it does look very much, when you compare between countries, seroprevalence rates at the moment are somewhere between 5 to 15%. Maybe New York has some of the highest seroprevalence in a city, but even they're just getting up to 20%. So despite all of the infection, all of the suffering, the number of people that have been infected, I think, is going to be worryingly and somewhat troublingly low compared to a population size that we have in the world. And just one last point, that sort of impinges very much on vaccines and therapeutics. I mean, we're very much hoping and seduced by the possibility of a vaccine quickly, and that it will be a vaccine that has a good efficacy with hopefully a single shot. But the reality is the vaccine world is priming with the first dose of vaccine and then boosting with a second. And with the population size that we need to look at globally, every time you need one extra dose of the vaccine, the logistics become fiercely complex very quickly.
Thank you, Professor Callum. That's very clear. Now let's close up the panel discussions with some questions from the audience. For those who wishes to ask questions in the audience, please type your questions in a box. And for those who wishes to voice up, please raise your hands virtually. No, that's always
Yeah, we already got some questions here.
Okay.
Yeah, let's go through that. I think.
So the first question, this is from Cameron Taylor. Even if a vaccine is successfully developed for COVID-19, is there any work being done on a universal vaccine for coronavirus for different strains of virus? So basically the question is, if we got a vaccine, is it one-stop shot forever? All the other coronavirus, future or past, will be vaccinated. Professors, anyone? Professor?
I think that's
Yeah, Callum, go ahead.
I was going to say, well, this isn't the only coronavirus of humans. There's 4 endemic coronaviruses that we have as seasonal coronaviruses, and they're quite distinct in terms of their genetics. There was some evidence early on that cross-reactivity between SARS-1 and SARS-2 serum exists, but I think it's a little bit of a push to say that vaccines will protect from all of these coronaviruses. In my view, this becomes much like looking at the world of influenza, where you have different coronaviruses that are able to infect humans. Some are worse than others, and the possibility of a universal vaccine, to my mind, is quite remote now. The good thing, though, is the universality of the platforms. And if some of these new platforms really do work, the RNA and the DNA delivery platforms, the ability to swap in new modalities, new spike proteins with the knowledge of a clinical efficacy and safety from SARS-2 perhaps accelerates that. But I personally find a universal coronavirus vaccine with the diversity that we see in the coronavirus world hard to imagine at the moment.
So this is good to have, but for now, we just need one. It's universal or not, right? So thank you, Professor Callum. Let's go to a second question. This is from Luis Abu Amak. what challenge does COVID-19 present to genomics in light of the genetic diversity, especially on African continent? Professor Callum, again, I think you are present a lot of the sequence information.
Yes.
Okay.
So if we talk about the genetic diversity of the virus, then I think most of us expect at the moment that the virus will continue to spread in the human population with sporadic variation and hopefully probably little in the way of directed mutation should change dramatically the phenotype of the virus. I think as immunity starts to build in the population, then the selective pressure becomes on the spike gene and possibly some other genes as well. And when that occurs in other coronaviruses, particularly coronaviruses in animals, we see genetic variation and the development of strains that are less neutralizable by the previous season's serology or 2 seasons ago. We also see recombination in coronaviruses, and that is something that we need to keep an eye on, I think, although I think there's very little evidence of recombination between the coronaviruses that already exist in humans. And the third thing, I think, is the thought process behind coronaviruses and how they may or may not manipulate the initial immune response so that the antibody tone, the antibody titer wanes over time, therefore making people perhaps vulnerable to reinfection. So this is going to be complicated, and really the more replication you have, the more chance you have of genetic variants, but you do need a selective pressure for them to then come to the fore. And at the moment, the virus is doing just fine without any real selective pressure.
I would agree on that point. Based on the genetic analysis of 10,000s of more sequences submitted to the gene bank, we really don't see tremendous selection pressure, i.e. that specific mutation that will drive away from the antibody of zero neutralization. So Dr. Callum is very correct. He says the tree that he demonstrated in early, really the sporadic ones, doesn't seem to have a significant consequences in terms of antibody recognition. But we have to remember this is an early epidemic. We're only learning from where we're able to get the sequences from. So hopefully in the future, virus wouldn't change as much as like influenza or HIV. So we have to shift gears every now and then.
Well, let's hope so. The third question from Xinhua Fen. I'd like to ask David and Lin-Chi. I'm curious if the crystal structure or antibody sequence have shown any common epitopes on the S protein. Can you improve the affinity and efficacy for the neutralizing antibody?
Dr. Ho goes first.
Dr. Ho?
Yeah, so for a couple of our antibodies, we are waiting for cryo-EM structures to come out, probably in the next few days or a week. From that, we could see if certain amino acid substitutions could result in even tighter binding. But we're already working on the structure of antibodies with nanomolar affinity. So it's not likely to go much higher than that. There are not too many antibodies with higher affinities than that. But nevertheless, we will be looking at that in the very near future with the idea of whether there are certain manipulations that could be made to improve the product profile.
Better and better.
Yes, we have a good number of antibodies that are able to neutralize SARS-CoV-2 in vitro. And then through collaboration with Professor Xinquan Wang at Tsinghua University, and he's a crystallographer, and also the team from a whole team from Tsinghua University and also Xihu Westlake University. We are in the process of resolving a good number of antibody structure to understand their structure basis for neutralization. the common feature that we have seen so far is that those antibodies capable of neutralizing that have a great power for competing with the ACE tool for binding of RBD. So basically push away the receptor and the RBD binding. That's really demonstrating their capacity in neutralizing. But as Dr. Ho just pointed out, some of antibody may show weaker neutralization, whereas the others is really showing higher neutralization. So we are in the process of analyzing the anatomic interactions between the antibodies and RBDs, so we could mutate some of those genes, some of the residues, so we could improve their potency and bindings. So those are ongoing. And then the newer ones, I'm sure, will have much higher binding and neutralizing activity.
Great. Thank the audience for raising these great questions. I actually wanted to continue on this, but unfortunately, we're limited by time. So let's end today's panel discussions with hope. Let's hope that a cure, whether it's a chemotype or a neutralizing antibody for COVID-19 will soon be identified. Let's hope that a vaccine will soon be developed and accessible to all. Let's hope that the world will soon be reopened and back to normal. With this hope, I wanted to thank Professor Ho, Professor Levine, Professor Zhang, and Professor Callum for your generous time and sharing. And thank you all for attending. Let's also thank Ali Health for providing a stage. Lee, back to you.
Okay. Yeah, I'm sure there are a lot of other questions. We do see, a lot of questions coming from the audience. But sorry, we have limited time. Especially, Sharon's time is already midnight already, right? So thank you very much for everyone, your precious time and participation. I hope you find the session as illuminating as I did, right? So I would encourage all of you to visit our website, the GMCC website, which is GMCC.alibabadoctor.com. Yeah, the website includes many resources on managing COVID-19, including prevention and control manuals, videos by doctors on COVID-19 topics. We will upload today's webinar to the website shortly because I do see there are a lot of audience asking whether we can get the PPT or notes. So we will upload shortly. And also you can find our other GMCC live sessions as we have today. So we have other 13 GMCC live sessions. You will find it there. In addition, we will be adding research articles to our website soon. So do come back to have a look. Thanks again to our distinguished speakers for your great insights. Thank you, Dr. Zhou, for your excellent moderation. And thank you all to our guests for your participation. Okay, good night. Thank you.
Good night. Thank you.